We're continuing on with the cell cycle. We've learned about interphase and that in the synthesis portion of interphase, DNA is duplicated. Also during this phase, the centrosomes are duplicated. And at the G2 mitosis, G2M transition, the centrosomes begin to move away from each other towards the poles. So here we see an animal cell and the nucleus with the chromatin, a thread-like material of the DNA, and the nucleolus. Here we have two centrioles. The centrioles are at right angles to each other, and two centrioles make a centrosome. The two centrosomes are beginning to pull away from each other towards the poles. And remember, plant cells do not have centrosomes, but they have microtubule organizing centers, MTOCs. So in this transition, the centrosomes are beginning to move away from each other. Mitosis is the process of dividing the nucleus. So it's a continuous process, and scientists have divided this into 4 phases in order to talk about what's happening in mitosis. The 4 phases are prophase, metaphase, anaphase, and telophase. And one way to remember that is with the sentence, pro met Anatel. Pro met Anatel, and he was very impressed. Prophase, metaphase, anaphase, telophase. The first phase, prophase, is where the nucleolus disappears, the nuclear membrane begins to disintegrate, the chromatin condenses into the chromosomes and they thicken and shorten, and the centrioles are moving to opposite poles and spindle fibers begin to appear. The spindle fibers are made out of protein. They're starting to appear, and here you see the nuclear membrane beginning to disintegrate. That is prophase. Prophase is the longest portion of mitosis. Metaphase is the second part of mitosis. The chromosomes meet on the equator, or otherwise the metaphase plate. Here we have the spindle fibers forming, and that's known as the mitotic spindle. The centrosomes are on either end, they're on the poles, and the kinetochore of each chromosome is attached to the centrosomes by a fiber. So that's where they're connected, right at the kinetochore. This is known as the mitotic spindle. The chromosomes, they line up single file along the metaphase plate. Anaphase, pro-met-ana, is the next phase of mitosis. Here, the kinetochore has been duplicated and the chromatids are separating, and each is now a separate chromosome. Before there were 2 chromatids to make the chromosome, now they are separate chromosomes. The telophase, pro-met-anatel, telophase, the chromosome clusters are surrounding the centrosomes, and you see here the nuclear membrane is beginning to form around each cluster of chromosomes. The supercoiled chromosomes unravel and become chromatin once again, and the spindle fibers are beginning to break down. Mitosis. Pro-met Anatel. They were stopped at the cytokinesis checkpoint. It was time to get ready to divide.